All right, so all the speakers are here from the last session, still? Because we're going to take questions for them. Yeah, we ready?
Yes.
Good. Questions? Amy.
This question's for Carly. Sorry, Carly, I did ask you at lunchtime. What do you think is mediated? I mean, I thought those hippocampal pictures that you showed us were amazing, showing that there's expansion, which we're seeing sometimes in our rodent models, and we possibly are seeing that in the post-acute and then follow-up imaging in humans. So you showed glial expansion contributing to the hypertrophy or increase in volume, but then you showed that neuronal CA1 and CA2 or CA3?
I think it was CA1
and 3, which is they're the vulnerable areas. They're the areas that are down in hippocampal sclerosis, which is very interesting. So what do you think the mechanism is?
Cytokine signaling. So I really think that when microglia become activated, there's a whole array of protein signaling pathways that are also activated. I think if they're traversing through the corpus callosum, they're actually setting off some pretty strong signals. And that, part of their immune response is to phagocyze debris, remove debris, and to fight infection, etc., as part of the evolutionary approach to maintaining CNS integrity. And I think that that's what we're seeing here is a signaling effect. Yeah.
And why the hippocampus if it's uninfarcted?
Yeah, so I think that that's the thing about secondary injury and inflammation. You actually don't have to have injury in a particular part of the brain to get an inflammatory response. And so that's what we're seeing in the contralateral brain. Previously, I actually published on a whole array of proteins that are differentially expressed on the contralateral brain, in the contralateral brain, and in actual fact, what I didn't present today, but there's a whole host of proteins that came up in that Fazil paper that were all contralaterally expressed. So specific proteins increased in the contralateral brain that were modulated by Fazil. So I don't think it's just about injury, but I think about it as how the whole brain talks to each other and the signalling effects and the power of protein signalling.
I had a question for Jess. Hi Jess. Nice touchscreen data. So in terms of the touchscreen data, the interpretation, do you think it's possible, the nature of the stroke, that there's an impact on visual acuity or head orientating towards the visual stimuli and or motivation and salience to the appetitive cues?
Let me think about that for a second. So I think they're all, this Tony, as much as I do, but I think they're all the immediate thoughts that come to mind. In terms of the visual acuity, what's interesting is this data that I've shown you, and you haven't seen is, we're using, it's just location. There's no actual discrimination in terms of perceptual features. The attentional task that we do does have those elements to it. And we're seeing a similar pattern thereby. I don't think it is purely just. We haven't done a visual acuity test in these mice as far as I know. Maybe others would know whether visual acuity has been tested after MCO. But my feeling is it's not that. And again, you saw my example when we did it right at the end where both it's just responding to one stimulus and they're able to do that in the same way. Your comment, we touched on this, I think when we had one of the last symposia earlier on at the Florey last year about motivational processing. Now I think that's something that's really emerged as part of our data that it's only until we've seen this data that makes us think actually that's a component that we should look after. There's been a heavy, you can imagine in terms of stroke, animal models of stroke, the motor component is something that I was very careful in making sure that we had measures put in place that allow us to detect that. But the motivation, what we want to do is potentially do another series whereby we did specifically look at whether it's whether it changes those affective changes that I alluded to. And to be honest, when we think about stroke and the clinical impact, it's likely that may actually be the case. But one of the things that's clear from our data, which I think is quite nice, is that we can at least dissect. It's not one of the things that comes out, and I know that potentially a lot of the work previously has been in things like the water maze, when you've got an animal that's motorically impaired and you put them in a water maze where they have to swim, the latency, there's a change in latency, you can't really be sure that's a cognitive component, not because they're trying to swim there. And so we went into these set of studies being very acutely aware of wanting to control for the motor components, which I think we have very nicely. But then what it's done is it's opened up the fact that there are these other components of the, you know, in terms of the features or the symptoms that we do have. So I think it potentially has elements of motivational or, you know, as you say, salience associated with the cue that may be important.
Okay, thank you. Yeah.
Jess, I have a question too for you.
Sorry,
Just going back to you, there was a question about latency again, just looking at the two graphs that you had. So correct me if I'm wrong, but with time, the latency decreased for correct responses. So the latency sped up.
across our test.
Across the
Are you
yes.
talking about in the sham or the...
Compared to the sham. So the sham was unchanged, but the stroke,
Yes.
the latency decreased.
Yeah.
But if I remember correctly, the inaccurate choices were still as bad as earlier on. Why, can you hypothesize why the latency decreased, but the error rate was still the same?
Yeah, so I must say, we've only just started dissecting these data, so I hadn't looked at the incorrect and correct and necessarily given them the same importance that you have. I sort of looked at both of those and saw that there was an impact of stroke, for example. But what it does say, and I think you rightly highlighted, is it's dynamically being impacted, it's changing across the longitudinal nature of it, which I think is really interesting. Now, you can imagine, and that's what makes me think it has elements of either processing speed or motivational. You can see how that, you can change the latency in which you make your responses, or want to make the responses, that are independent of whether you get them necessarily right or wrong. And so I think the accuracy, what's interesting is I think, remembering the accuracy data, that stayed pretty much the same, i.e. they were still getting it, you know, wrong right up until 48 weeks post. Whereas when you look at things like the latency, especially at 48, it would really come down. So this is what we would see, I think, in that it's telling us something about processing, underlying processing in terms of making a response, but that doesn't necessarily mean that it's impacting the animals know whether it's right or wrong.
Very nice talks in this session and throughout the day. My question is for Dr. Carlin. Sorry again, we have discussed this in the lunch break, but I thought it would be nice if everyone hears her insights to this. And my question is related with parasites, and you have nicely discussed how all the different cells are quite important. You have astrogliosis, and you have astrocytes glia, you have angiogenesis, and would looking at parasites in terms of either recovery or their contribution to the progression of the stroke or recovery be important in that mix of cells?
Well, I'm a firm believer of the vascular approach to dementia and neurodegenerative diseases. So when we think about changes in crosstalk between neurons and blood vessels, how astrocytes are involved in that, but neurons, but blood vessels actually don't only react through changes in parasites and endothelial cells engagement and as well as smooth muscle. So I don't know as much about parasites as I do about astrocytes and microglia, but what I do know is that there's a lot of work that's looking at targeting parasites to maintain collateral flow after stroke, and we know that people who have more flow, collateral flow in the stroke affected hemisphere tend to do better unless they develop malignant stroke. And under that circumstance, there's quite a significant drop in collateral flow over a short period of time. So there's probably a really big role for parasites, because as parasites activate, they constrict. And then when they've died, they retain that constricted form, which literally shuts the blood vessel down. So what parasites might do in terms of long-term after stroke, in terms of cognition, in terms of cognitive decline, it's a good question, and I think it really is worth investigating.
Well, I'll ask the last question to wrap up, and this is to all the speakers, whoever wants to answer. Most of the experiments that were shown involved lesioning young animals. And is there any evidence that older animals are more severely affected by the lesions? And also, wouldn't it be more appropriate to use older animals, considering that older people are more likely to be struck down by stroke?
Well, if we could get funding to do older animals, that's exactly what we would like to do. 3 years running in panel review, I got criticised for the cost associated with wanting to use older animals. But yes, I think that it would be really important that we should be looking at this all again in the older animals.
Thank you. It is impossible, so you need to kind of slip it in somewhere. We are going to be doing this in our aged animals. One of the issues is, and Vanessa has done some work are you Vanessa there? In aged animals, and one of the big criticisms is they'll die if you infarct them when they're old. But you found that the mortality is increased, but you can still do it.
Mortality's increased.
Yeah, so the mortality was increased, the infarct volume was bigger as well. But again, that was only, I think we would have only kept the animals alive for 24 or 72 hours. So given, like, for our sort of experimental protocol, keeping the animals alive for 48 weeks after stroke would be very difficult. I mean, you can always reduce the ischemic period to actually give them a softer sort of ischemia. But it's definitely sort of an option. We used particularly young mice initially, which we then sort of regretted, and from then on, every sort of subsequent experiment has been with 12-week-old mice, which is not old, but at least it's considered adult rather than adolescent, which is what we used with the 6 weeks. And yeah, but I think definitely aged mice is important, and then we could just reduce the sort of timeline and keep them alive for 6 months and just, see what happens.
I think now we've got that 1 year data that we could definitely keep them alive for longer. I think that, and then the issue, I mean, I think we've sort of done proof of principle in the sense that, childhood stroke is not associated with long-term degeneration. So childhood and adolescent stroke. So it is that combination of ageing and vascular risk that seems to be mitigating or driving that neurodegeneration.
Can I just add to that? There is actually quite a bit of literature out there to show that stroke in the aged animal increases astrogliosis. So the expanding scar is more severe after a stroke in an aged animal than it is in a juvenile animal. So there is evidence to show that the inflammatory response after stroke is increased with age.
Oh, very interesting. Good. All right. Well, I think we'll stop there. Did you, Jess? Yeah.
I just want to say, Ashley, you're absolutely nailing the point, but one of the things to just highlight maybe in terms of the various presentations presented is that this is about pushing the field forward. So when we started some of this work, my understanding was that traditionally with something like the MCAO, it was done in younger animals. I mean, whether it be a funding related issue or just because that was what we started with, I think what we're now seeing is more awareness. I mean, our studies with the longitudinal are pushing that out to say this is important. So I think gradually with time, it will start to ask. It's about asking and using the right models. But collectively, we need to push it that way. The other thing that I thought Tony would maybe comment is, even when we talk about aged rodents, there are other factors to potentially consider, which is a standard laboratory animal that sits in its cage and eats chow, and we look at them and they do rapidly gain weight. Is that the right model? So all of these things have positives but also negatives, and I think it's for us as a community, like in all scientific questions, to be considerate of those and then push the boundary.
Very good. All right. Well, thank you, everyone, and I will now hand over to Amy.
So again, thank you so much for that prior session, considering you were all ketotic and starving, but it kept you all awake, you see.